Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in this particular module, we are discussing about the cellular communications. So far what we have discussed about the basic mechanism of the cellular communications, what are the different components that are required for the cellular communications, and what is the relevance of the cellular communications in the previous two lectures. Uh, we have also discussed about the different types of mediators. So we have discussed about the animal hormones. So in the previous lecture, if you recall, we have discussed about the different types of animal hormones. So we have discussed about the peptide-based animal hormones. We have discussed about the steroid-based animal hormones. And then we also discussed about the special class of animal hormones. When we were discussing about the animal hormones, we discussed about the different types of hormones, their signaling mechanisms, and their relevance in terms of controlling the different types of activity. Now, in today's lecture, we are going to discuss some more aspects of the cell signaling, where we are going to discuss about the special signaling pathways which are operating in the different types of pathways. And then we also going to discuss about the plant hormones. And at the end, we are also going to discuss about the methods, how you can be able to study the cell signaling uh, using the different types of techniques. Now, talking about the cellular pathways, there are multiple pathways which are important for the where the cell signaling is very important, because cell to cell communication is very important and very important events for coordinated actions of the different types of cells. Now, one such pathway is called as the epithelial mesenchymal transition, or EMT. So, EMT is a very important pathway which is required for the cancer cells to move from their site of origin to the different sites within the body, right? And these kind of events are very important for the cell to gain the moment, and that's how they actually can go from one place to another place. So we'll start the lecture with the discussion about cell signaling in cancer cells. Especially, we are going to discuss about the EMT, and then subsequent to that, we are also going to discuss about the different types of plant hormones or cell signaling in the plant system. So as you can see, the cell signaling in the cancer cell, especially what is required for EMT. So the full form of the EMT is the epithelial mesenchymal transition, where the cells which are present in the epithelial type will convert into the mesenchymal type. And there is a significant difference between the cells which are having the epithelial morphology versus the mesenchymal morphology, because the epithelial cells are non-movable. They are actually be part of the different types of cellular structures, whereas the mesenchymal origin is allowing them for the movement, right? So they can actually be able to move from one place to another place, and that's how you have seen that the cancer cells are going to move from one their place of origin to the different places. For example, they can be, there could be a primary tumor which is going to be formed in liver, for example, right? Or lungs, for example, right? And then from liver or lung, it is actually going to move to the different cellular destinations. For example, it can move to the brain, it can move to the stomach, it can move to the other parts of the brain, right? Other parts of the body. So basically, the when you talk about the EMT, the cells are going to be in the epithelial type, and then they will actually going to form the mixed population of the epithelial and the mesenchymal cells, and then ultimately they will get converted into the mesenchymal type so that they can gain the the apparatus for movement, right? And that's how they will enter into the blood vessels, and then they will actually going to reach to their next destination. And in the next destination, they are actually going to do the reversal, which means they are actually going to get converted from the mesenchymal type to epithelial type, and that's how they are actually going to have the met transition, and then they are actually going to form the secondary tumor, and that's how the secondary tumor is going to be established in the next organ, right? So this is just a simple example how the EMT and the MET, which is actually the reversal of the EMT, is going to participate into the establishment of the tumor from its primary site to a secondary site. And this is basically the major reason why the cancer cells are very difficult to treat, or why there is a cancer cells are so dangerous, right? Because they can go from one part to another part, and that's how they can actually be able to infect the different parts of the body. And according to this movement also, the cancer could be of different grades, right? So EMT is driven by the transcriptional programming, right? So this is actually a transcriptional programming which will convert a epithelial cell into a mesenchymal cell by the several signaling pathways. For example, you have the TGF beta pathway, you have the BMP pathway, you have Wnt and beta catenin pathway, you have a Notch pathway, you have a Head-Hodge pathway, and the receptor tyrosine RTKase pathway. So all of these pathways are being activated when there will be a transcriptional programming, and as a result, the epithelial cells are going to be get converted into the mesenchymal cells. So these pathways are responding to the signals, right, from the tumor microenvironment, such as the growth factors, cytokines, hypoxia, and the interaction with the extracellular matrix or the ECM, right? And as a result, it is going to initiate the reactions of the EMTs. So we're going to discuss about the TGF beta signaling, right, as an example within the EMT. But apart from TGF beta, you're going to have all these pathways which are actually going to operate simultaneously or in different pathway, different cancer cell types, and that's how they are actually going to be responsible for the EMT pathway. So in a TGF beta pathway, upon ligand binding, the TGF beta R2, which phosphorylates the TGF beta R1 and its GS rich domain, creating a docking site for the SAM2, SMAD2, and SMAD3. So this is what is here written, right? So you are actually going to have the TGF beta R2 and TGF beta R1, and when the TGF beta is going to be bind to these receptors, they are actually going to have the cascade of phosphorylation, where they are actually first going to generate a docking site for the SMAD2 and SMAD3. And as a result of this, the SMAD2 and SMAD3, these transcriptional factors are then going to be then phosphorylated, allowing them to form a complex with the SMAD4, right? And then this SMAD complex translocated to the nucleus to regulate the target gene expression. The complex formation and the nuclear imports are mediated by the conserved domain and the nuclear localization signals. To regulate the signaling integrity, the TGF beta also activates the inhibitory SMADs like SMAD6 and SMAD7, which blocks for further SMAD recruitment, right? So basically, you have the fine balance of the TGF beta mediated signaling, which is responsible for the EMT, that it is actually going to activate the SMAD2 and 3 and then SMAD4, and then they will form a complex, like, and then this complex will going to translocate to the nucleus so that it actually can delineate further downstream signaling. But on the site, it is also going to activate the SMAD6 also, and which SMAD6 is actually going to inhibit the SMAD2 by 3 signaling. So BMP signaling allows a similar mechanism, but utilizes the SMAD1, 5, and 8 instead of SMAD2 by 3. And TGF beta signaling is also regulated through the post-translational mechanisms, and ETQ ligase like the SMARF1 and SMARF2 targets the SMAD protein for degradation, thereby suppressing the pathway. So there is a very tight control of the TGF beta signaling so that it is actually going to do its job, and then once the job is over, then it is actually going to activate the inhibitory pathway, and that's how it is actually going to be regulated. Inside the nucleus, so once this SMAD2 by 3 and SMAD4 complex is going to enter into the nucleus, the SMAD complexes regulate the EMT by activating the transcription of the key genes. They can induce the expression of SNAV-I1 and also the interaction with the snails to repress the epithelial markers like the E-cadherin and oculodin. They also influence the other EMT regulators such as Dept and HMGA2, which then regulate the SNAI1 and SNAI2 and the twist. TGF-beta signaling also activate the SWIT2 and reinforcing EMT by repressing the E-cadherin, right? So remember that there is a significant difference between the epithelial cells and the mesenchymal cells by their, the cell surface expression protein like the E-cadherin and oculodin, and all these proteins are heavily expressed when they are actually having the epithelial phenotype. But when they are getting converted into the mesenchymal cells, right, or mesenchymal phenotype, these cells has to be repressed so that they should not bind to each other. They should not attach to each other. And because of that, they will get detached from their primary site and then they will get into the blood, and that's how they will actually reach to the next destination, right? Apart from that, there are so many other kinds of modification also which actually allow these cells to gain the momentum like or movements phenotype like their actin, myosin, all those fibrils also going to be developed much better than the epithelial cells. So this is just a simple example of how the signaling, a very complex signaling, is regulating a very important pathway like the EMT pathway. So far what we have discussed about the cell signaling, right? And we have discussed about the cell signaling in the animal system, where we have discussed about the hormones, we have discussed about the different types of receptors and so on. And now we are going to start the discussion about how the cell signaling works in the plant system. Remember that the plants are also equally responsive as the animal, right? Many people have a perception that the plants are not very responsive compared to the animal because of the simple reason that when you are actually going to do anything to the animal, they actually show the quick action. They are actually going to show a movement. They are actually going to respond to your actions and so on, right? But that's something which doesn't happen in the plant because the plants are immobile. Plants are not going to show you a movement, except that if you are going to touch the mimosa pudica and those kind of plants, they will actually going to show a movement, and that's how you presume that they are actually responding. But the plants are also very highly responsive, and they are also having a well developed cell signaling. So plants are actually having a well-developed cell signaling for regulating their growth and development. They are also very well developed for responsive to the environmental stimuli. Remember that the plants are much more exposed to the environment compared to the animals because animals are having their own house, their niche. So because of that, the animals are pretty much protected from the environmental stresses. Compared to that, the plants are always been standing in the soil, and they are also going to be encounter different types of environmental stress, whether it is a drought, whether it is a high temperature, whether it is a low temperature, hypoxia, all sort of things, right? And then it has also been required the different types of molecules so that there will be hormonal communications. Then they also required because of for the defense also. So remember that the plants are present outside, and they are continuously getting the threats of the different types of bacteria, viruses, fungi, also on, right? So they are also having a different types of cell signaling for the defense also. Then the plants are also having the coordination among the different organs. For example, it's also going to have a coordination between the active photosynthesis, which is occurring in the leaves, versus the plant conduction of the plant, the water from the root, and then flowering and so on, right? Then it also required the cell signaling for the adaptations of the different types of stress. So you have the abiotic stress, you have a biotic stress, and so on. And then it also required the cell signaling for the regulation of the reproductive processes, which means the formation of the ovum, formation of the in the pollen grains, and then when there will be a fertilization, then the post fertilization, there will be a cell signaling required for development of embryos, and then development of embryo into the fruits and so on, right? So, cell signaling is very important in the plant also, because it is required for them to get adapted for the different types of abiotic and abiotic stress. Now, talking about the cell signaling in the plants, right? Plant perceives and integrate a wide range of external and internal signals to coordinate the growth, development, and stress, right? So, these are the exogenous signals, these are the endogenous signals. In the exogenous signals, you are actually going to have the light, you are actually going to have the mechanical signals, you are going to have the, and then you are going to have the atmospheric humidity, you are going to have the other plants, you are going to have the soil nutrients, you have the soil water, pathogens, gravity, carbon dioxide, and so on. Within the light, it is going to be responsive to the quality of the light, which means what is the wavelength of the light. Then it is going to be to the quantity of the light, which means it is going to respond to the intensity of the light. Then it is also going to respond to the duration of the light, whether it is a low duration versus high duration, which means whether it is going to be 8 hours versus 16 hours a day. Then it is also going to talk about the direction, right? Whether the light is coming from the east side or whether light is coming from the west side. Then for the mechanical signals, so mechanical signal means some kind of force. So it could be like wind, right, which is actually going to, you have seen that the wind is also swinging the plant, right? Then it could be some herbivores, like for example, you have different types of cows, buffaloes, all sort of herbivores like deer. They are also attacking the plants, and they are also disturbing the plants, and they are causing the mechanical disturbance. And then you're also going to have the atmospheric humidity, right? You're going to have the other plants. Remember that the plants are also competing with each other for different types of nutritions. In some cases, the plants are also helping each other by providing the different types of nutrients and so on. So what plant is present in the proximity, that also is going to give them a signal. Then we also have the soil nutrients. So you're going to have the different types of nutrients present. So you may be having the macronutrients or the micronutrients and so on. Then amount of water present, that also is going to give the abiotic stress. Then we also have the biotic stress, like different types of pathogens what are present in the soil. Maybe they're good pathogens, or they are going to be good bacteria or bad bacteria. And then you also have the gravity, what is the height of the plant, right? Whether it is present on a plant which is present on Himalayas versus a plant which is present on the seashores and so on. And then we also have the signal for the the gaseous molecules like the carbon dioxide, nitrogen, the ethylene and all that, right? And then we have the endogenous signals. So you can have the growth regulators like different types of hormones like cytokinins, ethylene, gibberellin, auxin, abscisic acid. And, and then we also have the mechanical endogenous signal like growth related tissues, compressions and tension. Then we have the defense signals like the jasmonic acid, salicylic acid. Then we have a developmental regulators like the mobile RNAs. And then we also have the metabolites like the sugars, glutamate, and so on. And all these, whether it is the exogenous signals or the endogenous signal, is going to regulate the growth and the development. So let's first start with the first signal, and that is the light signal. So light is a critical environmental cues that influence the plant growth, development, and behavior. Plant perceive the light through the specialized photoreceptor that detect the light HST, wavelength, duration, and directions. Light signaling pathway transduce these signals to regulate the gene expression and coordinate the physiological responses. Key developmental processes influenced by the light include the seed germinations, photophoresis, shade avoidance, and the flowering. Now, what are the different types of photoreceptors? So, photoreceptors are the specialized protein that absorb the light and trigger the photoresponsiveness in the plant. Most constant of a plant protein linked to a light absorbing chromophore except the UR8, which include functions without one. They are sensitive to the light wavelength, intensity, and the photoperiod durations, allowing the plants to adapt to the changing environment. Photoreceptors regulate the development and protective responses through a plant's life. What are different types of photoreceptors? So there are different types of photoreceptors and their light sensitivity. So you can have the phytochromes, which is red and far red, and it is going to absorb in the range of 620 to 800 nanometers. Then we have the cryptochromes and phototropins and zoot lights family, which is actually going to absorb in the range of UV, so UVA and blue light. And then you also have the UVR8, which is actually going to absorb the UVB light, which is in the range of 280 to 320. So this is all these photoreceptors are mostly being present onto the leaves, and they are actually going to absorb the light of the different wavelength. Like they will be absorbing the UVB, which is range in the 290 to 315, then UVA in the range of 320 to 390. Then you're going to have blue light, which is in the range of 390 to 500. Then you're going to have a green light, which is in the range of 530 to 570, and red and the far red. And for all these, you are actually going to have the different types of photoreceptors. And all these photoreceptors are actually going to delineate the downstream signaling, and that's how they are actually going to control the seed germination, seedling deauriculations, leaf development, flowering, and senescence. So basically, the primary source of the light is the sun, and the sun is actually going to regulate the all photoreceptors mediated responses. So this is an example of light signaling. Is a red light induce the seed germination mediated by the photoreceptor phytochrome B. So in Arabidopsis thaliana, which is a sample plant, five phytohormones like phyt, phy2, phyt have been identified, while in some monocots such as rice and maize, the three phytochromes like phy A, phy B, and phy C are commonly found. While the phi A is primarily responsible for the far-red light absorbance, phi B is the main regulator of the red light induced seed germinations. Now, what is exactly happening, right? So, seed germination is regulated by a balance between the two key hormones, the abscisic acid induced dormancy and the gibberellinic acid promote germination, right? So under the red light, the phi B is converted from its inactive pro form to active PFR forms, which translocated into the nucleus and cause the degradation of the transcription factor, which is called as phytochrome interacting factor 1. So when there will be a red light, which is going to fall, it is actually going to activate the both of the photoreceptors, and basically the phi B is actually going to get converted into the phytochrome interacting factor 1, and then phytochrome factor 1 is actually going to inhibit the production of the abscisic acid. So it is actually going to inhibit the production of the abscisic acid, and at the same time it is also going to enhance the production of the gibberellinic acid through a receptor or GO3-OX1, and as a result it is actually going to promote the seed germination. But at the same time, when there will be the red light, or then that red light is going to be absorbed by phi B, and it is going to form the PFR1, and that is going to inhibit the whole reaction. And that's how there is a fine control of the light induced seed germinations. So PFA1 acts as a repressor of the seed germination by suppressing the GA biosynthesis by the repressing of this and that, promoting the GA catabolism by activating this, and promoting the ABA synthesis through upregulation of ABA1, NCD1, and light, inhibiting ABA catabolism by repressing the type 3, right? Now, thus degradation of PF1 leads to the increased GA level and reduce the ABA level, thereby lifting the dormancy and promoting the seed germination. Under low light or far red radiation, phi B is converted back to the inactive PFR form, and the PF1 accumulates and seed germination is repressed. Then we have another signaling which is called stress-linked signaling. So you have two different types of stress. You have the biotic stress, you have a abiotic stress. So biotic stress like the infection by the virus, bacteria, fungi, nematodes and attacks by the insects or the herbivores. Whereas the abiotic stress, you have the droughts and flooding, which means the low water or the high water. Then you have the salinity, you have the heavy metals, you have a UV radiations, you have a nutrient deficiencies, you have extreme temperatures, right? So all these and plant constantly face the challenging environmental conditions that can negatively affect their growth, development, and the productivity. These stresses are broadly classified as the abiotic or the biotic stresses. The exposure to such leads to the rapid physiological and molecular changes affecting the processes like the photosynthesis, respiration, flowering, senescence, and gene expressions. To survive and adopt, plants have the evolved complex network that perceive these stresses and trigger the appropriate defenses and tolerance responses. Then we'll come back to the plant hormones. So that is very very important that plant hormones are also the the good mediator of the cell signaling, and that's how they are actually going to regulate the different types of events. Like plant hormone or the phytohormones are the organic signaling molecule that regulate the plant growth, development, and the adaptation. They act at a very low concentration, coordinating the cellular responses to both the internal and the environmental stress. They are typically synthesized in one tissue and transported to other. They include the classical hormones such as auxin, gibberellin, cytokinin, abscisic acid or ethylene. And then they also have the newly discovered hormones like the basinosteroids, jasmonates, salicylates, styrolactones, and nitric oxide, polyamines, and the peptides. So these are the new molecules which are being also found to modulate or cause the downstream signaling, and that's how they are actually going to modulate the activities. So there are five classical plant hormones. You have a auxin, cytokinin, gibberellin, abscisic acid, and the ethylene. So let's first talk about the auxins, right? So auxins are indole-based plant hormone derived primarily from the tryptophan. They regulate the cell divisions, elongations, and differentiation, influencing the identity and the behavior of a plant cell. Auxin is primarily synthesized in the shoot apical meristems, leaf primordia, young leaves, and the developing seeds from where it is transported to other tissues to regulate the growth and development. It is crucial for processes like the phototropisms and gravitasms, where uneven oxygen distribution drives the directional growth in response to light and the gravity. There are natural oxidants compounds. So you have the indole acetic acid, you have the indole butyric acid, you have the 4-chloroindole 3 acetic acid, and then you also have the phenyl acetic acid. So these are the four compounds which are natural oxidants which are found into the system. And all these natural compounds are actually having aromatic ring because they all are being derived from the tryptophan, right? So they are being derived of the indole and containing a carboxymethyl substitute. So this is the structure of the IAA, this is the structure of IBA, this is the structure of PAA, and this is the structure of 4-chloroindole acetic acid. So you have the indole acetic acid, you have IBA, you have a 4-chloroindole acetic acid, and you have the PAA, and this is the molecular formula, right? And this is the structure. Now, how the oxygen signaling works? So, oxygen signaling in plants is governed by the three core components, TIR1 or AFB, so transport inhibitory protein responses 1, or the oxygen signaling F protein or the oxygen receptor. So, you're going to have the TIR1. So, in response, in absence of oxygen or the presence of oxygen, the TIR1 is actually going to, so oxygen or indole-acetic acid transcriptional co-suppresses. So ARF oxygen responsive factors, the transcription factors, and under low oxygen conditions, the TIR1 or AFB show weak affinity for the oxygen or indole-acetic acid, allowing the oxygen or indole-acetic acid to bind the ARF and represses the oxygen responsive gene expression. So basically, the oxygen is going to bind the TRF1, which is actually going to show the weak affinity for the oxalin or the indolestic acid, and that's how there will be an oxalin responsive factors. They are actually going to respond to the oxalin, and that's how they are actually going to change the transcriptional activities within the plant. As oxygen level increases, the oxygen binds to a pocket in TRF-AFB protein, acting as a molecular glue that enhances their binding to the oxygen or indolactic acid. This leads to the polyubiquinylation and degradation of the oxygen and release of the RF, which are then free to activate or represses the target genes. This mechanism enables the oxygen to rapidly and precisely regulate the gene expression in response to the developmental or the environmental cues. Now, what is the function of the axons? So, apart from the other type of functions, the axon is very, very important for the phototropisms and the gravitropism, right? So, phototropism means the responses toward the light, and the gravitism is the responses toward the gravity. So apical dominance is mediated by the downward movement of the auxin from the shoot tip, which suppresses the growth of the auxiliary beads. So this is one of the important factor of the auxin. It promotes the cell elongation in the shaded stems or the shade avoidance. Then it stimulates the flower development, setting and ripening of the fruits. And a high oxygen level prevents the leaf or the fruit drops. Declining the oxygen permits the ethylene to trigger the senescence. So this is exactly what happens when there will be a sun, right? So if the sun is coming from the above, right, the plant will grow in this direction. But if the sun is coming from the this side, right, then it is actually going to ask the accumulation of the oxygen on one side. So wherever there will be an oxygen accumulation, it is actually going to allow the movement of this more. So it will actually going to allow the growth of this portion. So there will be a more elongated cells. It is going to be on this side. And as a result of this, the whole plant is going to move towards the sun side. So if you have a sun from one side, the sun causes oxygen to redistribute away, leading to its accumulation onto the shaded side, right? So basically, it's this side you have a sun, right? So it is actually going to accumulate onto the opposite side, and because of that, it is actually going to allow this portion to move further or move larger compared to this portion, and that's how it is actually going to bend. Then we'll talk about the next hormone, and the next hormone is cytokinins. So cytokinins are a class of adenine-derived plant hormone that primarily stimulate cell division and influence various aspects of growth and development. Synthesized mainly in the root, they travel upward to the xylem to reach the target tissues like such as shoots, leaves, and fruits. Cytokines are crucial for regulating the cell differentiation, shoot initiation, delay of senescence, and the overall plant morphogenesis. Site of cytokinin biosynthesis and occurrence include root tips, developing seeds, young leaves, stems, flowers, follicles, fruits, and shoot meristome. There are natural cytokinins compounds. So you have the transzetine, you have ciszetine, and you also have the isopentenyl adenine, right? So all these are actually the adenine derivatives. This is the structure of the cytokinins. There are different types of cytokinins, right? So you have in nature, the cytokinins are based on an adenine ring with a side chain of isopentenyl attached at the N6 position of the adenine. The presence of double bond in this side chain leads to the formation of geometric isomers, and zeatin can exist either in a transzetine or the ciszeatin form. So basically, this is the adenine structure, right? And then you're also going to have the isopentenyl chain attached to the amino groups present on the adenine, right? And that's how these are the some of the derivatives. This is the trans form, this is the cis form. How the signaling works into the cytokinins. So cytokinin signaling in plants begin when the cytokinin binds to the membrane localized histidine kinase receptor AHK2, AHK3, AHK4, and CRE1. So these are the receptors for the cytokinins, which are called AHK2 and AHK3, and these are transmembrane receptors. Upon binding of these receptors autophosphorylate on a conserved histidine residue and transfer the phosphate to a conserved aspartate residue. So when there will be a cytokinin binding, it is actually going to initiate the autophosphorylation, right? And the autophosphorylation occurs onto the aspartate residue. The phosphorylated group is then transferred to the histidine containing the phosphotransfer protein in the cytoplasm. So it is actually going to transfer the phosphate group to the HP1, and then the HP1 moves into the nucleus and phosphorylate the type B ARR1s, which act as a transcriptional regulator of the cytokinin responsive genes. This system includes a negative feedback loop where the ARR induced by cytokine signaling act as a repressor to modulate the responses. So there will be a feedback mechanism where the AHP is also going to initiate the repressive activities, and that's how it is actually going to down-regulate the cytokine signaling once the effects are or the requirements are already being completed. Then we talk about the third hormone, which is called as the gibberellic acid or gibberellins. So gibberellins are the large family of plant hormones that share a common Gibbon ring and exhibit diverse biological activities. The most well known among them is the gibberellic acid. Gibberellins regulate various stages of the plant growth and development, notably the seed germination and seed elongation, leaf expansion, and the flowering. Site of gibberellin biosynthesis and occurs includes the young tissue of the shoot and development seeds. These are the key bioactive gibberellins. So you have GA1, GA3, GA4, GA7, right? And gibberellins are large family of over 125 structurally related plant hormones, primarily synthesized in the roots and shoot, apical meristums, young leaves, and the developing seeds. This is the general structure of the gibberellic acid, right? So gibberellins are tetracyclic triterpenoid acid that exist in two structural classes, those with the 19 or 20 carbon compound. The biologically active forms are usually 19 carbon gibberellins, which feature as a five-membered lactone rings, include linking the C4 and C10 due to the loss of C20. The hydroxylation at C3 and C13 significantly enhances the activity of the gibberellic acid. Gibberellin act as a primary examples being a dihydroxylated 19 carbon gibberellins. How the signaling works? So gibberellin acid initiate the signaling by binding to the receptor which is called as GID1 or gibberellin insensitive DOF 1. So this is the GID1, right? In the presence of GA, the GA-GID complex binds to the del A protein, which are negatively regulator of the GI responses. This interaction triggers the recruitment of a ubiquitin ligase complex, marking the del A protein for the degradations. And once the DLA is degraded, right? So degradation of the DLA releases the key transcription factor and other regulatory proteins such as refoldins, enabling the activation of the GA responsive signaling. So once the DLA is being degraded because the ubiquitin pathway is going to be activated, it is actually going to activate the transcription factors, and the transcription factor is then going to activate the GA responsive genes. In the absence of GA, when the GA is not present, the del A protein remains stable and suppresses the transcription, thereby inhibiting the growth processes like seed germination, stem elongations, and the flowering, right? So when the GA is not present, the GID1 is actually going to maintain the DELA, right? And that is actually going to suppresses the transcription factors and the prefoldins. And that's how it is actually going to not allow any of these processes which are being governed by the gibberellic acid, such as seed germination, seed elongation, and the flowering. Now let's come to the next plant hormone, and the next plant hormone is the abscisic hormone, which is responsible for the dormancy. So abscisic acid is a plant hormone crucial for regulating the growth and the development. It controls the processes such as leaf senescence, dormancy, and the seed germinations. It is a plant stress hormone and plays a crucial role in plant responses to the environmental stress. Site of ABA biosynthesis and occurrence includes the roots, mature leaves, particularly in response to the water stress, and the seeds. The biologically active form of ABA in plant is the SABA, which has the two cysteine 4-trans side chains. The trans ABA isomer is biologically inactive. ABA is a 15-carbon cis-citrine and it is unique among the plant hormones for having a asymmetric carbon at position 1, giving rise to the two enantiomers RABA and the SABA. So this is the structure of the ABA, right? It consists of six carbon ring with an attached side chain, right? So this is a side chain, this is a six-member carbon ring. The natural occurring and biologically active form of a plant is the SABA. How the signaling works? Abscisic acid signaling. In the absence of ABA, the phosphatase ABA insensitive inhibits the SNRK2 kinases. So in the absence of ABA, the ABA11, which is a phosphatase, is going to inhibit the activity of this particular kinase, and that is in turn. So when the ABA is present, it is binding to the pyrobactin resistance 1 or PIR1. Pyrobactin resistance 1 like receptor, which then binds to and inhibit the AB11. So when the ABA is present, ABA is going to bind the PIR1 and that is going to inhibit the activity of AB11. And as a result of that, the kinase, the SNRK2, is going to be on an active state, and that's how it is actually going to activate the ABA responsive factors, and that is going to change the transcription. So this releases the SNRK2 from suppression, allowing them to activate the ABA responsive element binding factors or the transcription factors. These ABA regulates the expression of the multiple genes, which is mostly being responsible for the dormancy, or they are also being responsible for the withstanding the different types of stress. So they are actually going to make the plant withstand the different types of stress. So it is actually going to be ABA is going to generate all of a lot of protective signals, so that it will instead of plant spending its energy into the growth and other kinds of activities, it will go into dormancy, or it is going to withstand the stress by reducing the energy production and so on. Then let's come over to the next hormone, and the next hormone is the ethylene. Ethylene is also going to be called as gaseous hormone, right? Because the ethylene is a gas actually. So ethylene is a simple gaseous plant hormone that play a crucial role in coordinating the growth, development, and the stress responses. It is unique among the plant hormone due to its gaseous nature and act as a signaling molecule in many physiological processes. Site of ethylene biosynthesis and occurring includes the tissue undergoing the ripening, such as fruit, roots, shoots, particularly in response to the stress. This is a simple example of the structure of the ethylene, which is like C2H4, right? So this is the structure of the C2H4, which is ethylene. So ethylene is a simple unsaturated hydrocarbon gas that functions as a natural occurring the plant hormone. And what is the signaling mechanism of the ethylene? So ethylene signaling primarily occurs at the endoplasmic reticulum, where the ethylene binds to the receptors such as the ETR1, ERS1, ETR2, ERS2, and the EIN4, right? So ethylene is a gaseous molecule which then when it enters into the cytosol, it is going to bind the different types of receptors like the ETR1, ETR2, ERS1, ERS2, and EIN4. And in the absence of ethylene, these receptors activate the kinase which is called as CTR1, which phosphorylate and inactivate the EIN2, preventing the downstream signaling. So when there is no ethylene, it is actually going to activate the RAF-like kinases CTR1, and which actually going to inactivate the transmembrane protein EIN1 or EIN2 actually, and that's how it is actually going to inhibit the downstream signaling. But when the ethylene is present, right, so when the C2H4 is present, it is actually going to activate the EIN2 or downstream signaling. So and that's how it is actually going to have the activation of the ethylene responses genes, and that is actually going to regulate the plant growth and development, especially the fruit ripening. So fruit ripening is one of the important feature of the ethylene hormone mediated signaling. So when the ethylene is present, it binds to the receptor, leading to CTR1 inactivation. So this CTR1 is not going to be active. This allows the C-terminal end of the EIN to be cleaved and translocated to the nucleus, where it activates the transcription factors like EIN3 and EIL1, triggering the ethylene responsive gene expression. So this is all about the different types of mediators. So we discussed about the plant mediators in terms of the different types of hormones, abiotic factors, biotic factors, and so on. We have also discussed about the animal system, where we have discussed about the different types of hormones and so on. Now, the question comes, if there is a cell signaling, right? What are the different weapons, or what are different tools are available to study the cell signaling events? So cell signaling, there are different methods available to study the cell signaling, right? These are highly dynamic systems that constantly respond to a wide range of internal and external signals, right? Remember that the cell signaling is a process which occurs continuously, right? And that's why you require the system which actually can allow you to study these events. Talking about the methods, so we actually have the four different methods. We have the Western blotting, we have a phosphoproteomics, we have a microarray, and we have the reporter gene assays. The applicability and the context in which you can be able to use any of these methods are very, very different, right? In some cases, you can use the Western blotting. In some cases, you can use the phosphoproteomics. In some cases, you can do microarray. And in some cases, you can actually be able to use a reporter gene assay. We are not going to discuss in detail about these processes, procedures, and so on. But if you like, you can be able to study all these in one of my MOOCs course, which is called as the experimental biotechnology. So there's a MOOCs course already available, where you can actually be able to study the experimental biotechnology, where you can be able to study about the Western blotting, where you can actually be able to study about the reporter gene assays and so on, right? Phosphoproteomics and microarray is probably not present in this particular MOOCs course, but you can be able to study that from many excellent and good quality MOOCs courses are available onto the portal. So Western blotting. Western blotting is a widely used technique to detect a specific protein and analyze their expression or activation during the signaling events. You have the four different steps. You have the sample preparation, then you're going to do the gel filtration, then you're going to gel electrophoresis, then you're going to have the membrane transfer, and then you are going to have the immunodetections. All these processes we discuss in detail in that particular MOOCs course. So sample preparation, you can be able to do the sample from a single cell, you can be able to do the sample from a tissue, and so on, right? And there is a different types of methods available how you can be able to prepare the sample from the cell or the tissue. Once you develop the sample, then you actually can resolve that onto the electrophoresis like the SDS page, and then you can actually be able to do the membrane transfer, and after that you can actually be able to do the immunodetection, so where you can actually use the primary antibody, and you can also use the secondary antibody, and so on. So there are so many technical details available of these key events, like what are the different types of SDS page you are going to use, what are different methods for membrane transfer and what are the different types of immunodetections, because even within the immunodetections, you're going to have the blocking events, then you're going to have washing, then you're going to have the incubations with the primary, secondary, and so on. So all this is very extensive. So we are not discussing about all these events. Then the second method is the phosphoproteomics. So phosphoproteomics you are going to do when you would like to see the response of the global proteins present in the particular cell. So remember that the Western blotting is you are going to do for single or few proteins, right? So that is only when you know that which protein is getting affected. But if I want to know, for example, if I want to know if I add the ABA to the cell, what are the proteins, right? So if I know proteins, I can do the Western. If I don't know the protein, I want to identify all the proteins, then I should do the phosphoproteomics, especially if I want to know whether what are the proteins are going to be get phosphorylated. So phosphoproteomics is to do to looking at the global picture. Western blotting is going to do to see the specific questions or specific problems, right? So phosphoproteomics is a specialized area of proteomics focused on identify and analyze the protein modified by the phosphorylation. And since phosphorylation often marks when the protein is active or inactive, studying these events helps to understand the cellular processes. Unlike the basic protein expression studies, phosphoproteomics not only reveals which proteins are present but also indicate which signaling pathway may be turned on or turned off. One common method for studying the phosphoproteomics is the mass spectrometry, which enables the large-scale identification and quantification of the phosphorylation event across the proteome. So the key steps what are includes for the phosphoproteomics is that you're first going to have the sample preparation and then you're going to have a digestion. So cell or tissue are lysed to extract the proteins which are then digested into peptides, typically using the trypsin. Then these are going to be enriched for the phosphopeptides. So since phosphorylated peptides are usually low in abundance, the specific enrichment techniques like IMAC or the TI2 affinity chromatography are being used to isolate them. Then once you have done that, then you are going to do a mass spectrometry to analyze the sizes of those proteins. So then the enriched phosphopeptides are analyzed using the high resolution mass spectrometry to detect their mass and phosphorylation sites. And then you're going to have the data analysis. So advanced bioinformatics tools map the phosphorylation sites, quantify the changes, and identify a regulated pathway or the potential drug target. Phosphoproteomics enable the global analysis of the phosphorylation event that drive the intercellular signaling, and that is what the main purpose of doing the phosphoproteomics. You are going to see all the proteins present in the cytosol or different organelles. Since kinase modified the specific amino acids, typically like serine, threonine, and tyrosine, to regulate the protein activity, localization or the stability tracking these modification provide the insight into the pathway. So basically, it is going to give you the key proteins, what could be involved, right? But and then you can actually be able to do the Western blotting to identify and to verify these studies. One method uses the SILAX or stable isotope labeling by the amino acid in cell culture, a metabolic labeling technique where cell incorporate the non-radioactive isotopically labeled amino acid during the growth. And then after stimulation, samples are collected over time and analyzed by mass spectrometry to detect the phosphorylation changes across the many proteins. This reveals which pathway are activated and help to map the complex signaling network in response to stimuli. So phosphoproteomics is going to give you a global phosphoproteins. Okay. And once you know the global phosphoproteins, using this data, you can be able to draw the signaling pathway. You can be able to use that in conjugation with the Western blotting to reduce the signaling pathways. Then the next technique is the microarray. So this is the technique where we have looked at the global phosphoproteins, right? Now, if you want to know at the level of gene, then you can actually be able to use these. So this is actually going to tell you about the global gene expression, right? Data, and that also is going to indirectly support your results from the global phosphoproteomic data or from the Western blotting data. So microarray are high throughput laboratory tools to analyze the expression of thousands of genes simultaneously. They consist of a solid surface, usually a glass slide, on which the thousands of DNA probes are being fixed in a grid-like pattern. When labeled, the nucleic acid from a sample are applied. The complementary sequences bind to the probes, allowing to detect and quantify the gene expression across the genome in a single experiment. This technique can help to understand the gene activities, disease profiling, and the cellular responses to the various types of stimuli. In a typical microarray experiments, messenger RNA is extracted from the cell and converted into the cDNA, which is then fluorescently labeled. This process we have discussed in detail in one of my MOOCs course, which is called as the molecular biology, right? How you can be able to generate the cDNA from the messenger RNA, right? The labeled cDNA is then applied to microarray chip containing the thousands of DNA probes. Each probe corresponds to the specific gene. If the cDNA binds to probe, it indicates that the gene is being expressed in a sample. The chip is then scanned to measure the fluorescent intensity, which reflects the level of gene expression. So microarray can help to monitor changes in the gene expression across the thousands of the genes simultaneously, helping to analyze how signaling pathway affects the transcriptional activity. When cells are exposed to a signal like a growth factor, cytokines, or stress stimuli, the downstream transcriptional responses can be understood using the microarray, right? By comparing gene activity before and after a specific signal is introduced, we can see which genes are being turned on or turned off, and it figure out which signaling pathways are being active, how cells respond to different signals, and what might be going wrong in disease like the cancer. So, the major purpose of doing the microarray is that you want to study the different types of genes which are going to be upregulated or downregulated. Studying these data is going to help you to understand the signaling pathways being triggered in response to a particular type of stimuli. It could be a stimuli which is abiotic, or it could be a biotic, it could be anything, right? Then the last is the last method, which is called as reporter gene assay. So reporter gene assay is also be a confirmation of your microarray data. So once you have the microarray data, you can actually be able to use the reporter gene assays to confirm. So reporter gene assays are mostly being used to study the activity of the promoters, right? So remember that all the gene expression is being governed by the promoters. So if you want to know, if you want to study the promoters, the activity of the promoters, because that is only going to allow, tell you which gene is going to be upregulated and downregulated, you can actually be able to study the promoter activity using the reporter gene assays. So reporter gene assays are widely used to study the gene regulation and signaling pathway, and in this method, a reporter gene such as the luciferase, Dept, or beta-galactosidase is placed under the control of a specific promoter or regulatory sequence of interest, which means the promoter which you want to study, right? Whether this promoter is a strong promoter or weak promoter, whether it is going to increase the expression of the gene or downregulate the gene, and so on. So when the signaling is being activated, it leads to the transcription of the reporter gene, which produce a measurable signal. It could be a light, it could be a fluorescence, it could be a luminescence, and so on. This can allow to the indirectly monitor the pathway activity, assesses the reporter strength, and the identify responses to stimuli or to the drug. Key steps for one of the commonly used methods for, like, you can actually be able to construct. So a reporter gene, for example luciferase or GFP, is cloned downstream of a regulatory event or the promoter. Then you do the cell transfection. The construct is introduced into the cell culture using the methods like the electroporations, and then you do the stimulation. So cells are then treated with the signaling molecules to activate the pathways. Then you do the incubations, so cells are allowed to respond, leading to the transcription of the reporter gene if the pathway is active. And then you do the detections, so reporter activity is measured using the appropriate methods. You can actually use the luciferase, GFP, and beta-galactosidase. So you can use the luminescence, you can use the GFP, like the fluorescent imaging, or you can use the colorimetric assays. Reporter gene assays allow the easy monitoring of whether a signaling pathway is active or inactive. When the signaling pathway is active, it leads to the expression of the reporter gene, resulting in a measurable signal like the light or the fluorescence. The intensity of this signal reflects the strength or the activity of a signaling pathway. This approach helps in tracking the gene regulation and understanding how cell responds to the different types of signal. So far what we have discussed about the cell signaling, we have discussed about the methods how you can be able to study the cell signaling. So if you recall, in this particular module, what we have discussed about the relevance and the importance of the cell signaling. So we discussed about the different types of factors which can actually be able to potentially trigger the cell signaling, and how the cell signaling is regulating the different types of events, whether it is related to growth, whether it is related to withstanding the different types of stress, whether it is related to the delineating the death signals, or whether it is the activities related to the activation of the different types of caspases and so on. And in talking about the plants, we have also discussed about how the cell signaling is regulating the responses against the light, different types of biotic and the abiotic stresses, and then how the cell signaling mediated by the different types of plant hormones are also regulating the different types of downstream cellular events such as seed germination, growth, photoperiodisms, and the geotrophisms, and so on. So with this brief discussion about the cell signaling, I would like to conclude my lecture here. Subsequent lectures we will discuss some more aspects related to this particular course. Thank you.